Okay, good morning, everyone. Thank you very much for coming here and for being here so early, especially after the fact that we have a late start tomorrow. It is really my great pleasure to be invited to speak at ESI SyNC. I did start my independent research career in Frankfurt at the Max Planck Institute for Brain Research, where I spent 6 wonderful and productive years, and recently moved to the Technical University of Munich. So I have been hearing about ESI SyNC for many years, and it's really a great pleasure to finally have the opportunity to speak at this event. So what I want to tell you about today is some recent work in my group that deals with the question of how functional circuits and computations emerge in very early stages of development. So we're interested in how young brains develop, namely how the connections that are initially very inappropriate and imprecise transition to being obviously non-random and precise and give rise to functional circuits that can produce computations and behaviors that we take almost for granted in adulthood. So we know that there's multiple mechanisms that are involved in this process. Some of them are completely independent of patterns of activity, and they are determined by the interaction of different sets of genes and molecules. Yet others are dependent on patterns of neural activity. And normally, when we think of activity, we really think of activity as such that is generated by the sensory organs as we perceive the world around us. So through the different sensory modalities, audition, vision, olfaction, and so on. But in very early development, many of the sensory organs that receive and process this sensory information are immature, which raises the question then of where would neural activity come from? Well, it turns out that many developing circuits across species and different brain areas possess this temporary ability to produce special spontaneous activity completely in the absence of this sensory experience. And so here we see an example of such activity recorded in the brain of a preterm baby that was born 10 weeks prematurely. What you see at the top is that this baby doesn't really move at all. Nonetheless, in the recordings below, you can see that there are these events known as delta brush oscillations that are highlighted here in the gray boxes that demonstrate that the ability, in this case of the somatosensory cortex, to generate spontaneous activity in the absence of any kind of movement. We're a computational group, and we mainly work with experimental colleagues from where we analyze and study the spontaneous activity mainly in model organisms. And so, of course, we use the mouse as such model organism, and especially we study the sensory system, in this case, the visual system. So here, spontaneous activity is generated in the different circuits in the visual system, beginning with the first one that processes vision, which is the retina. And so here, many labs, prominently Marla Feller and others, have actually studied the spontaneous activity that is generated in the retina. So you see, this is a calcium imaging recording. The tissue that we see here is about 0.5 millimeter by 0.5 millimeter in size. And what we can see is this beautifully patterned activity. So these very rich spatiotemporal correlations that have been known now for several decades. This spontaneous activity, of course, propagates downstream in the visual system. So in the thalamus, the LGN, which is the visual area of the thalamus, the visual cortex, the primary visual cortex. And let's look at what activity looks like in the visual cortex. This, I should point out, is a slightly different scale. So this is several hundred microns in length. And what we see here is cellular resolution activity that, again, tells us that there is a lot of spontaneous activity also in the visual cortex. And this spontaneous activity actually has a little bit different patterns than in the retina. It doesn't seem to propagate in the form of these waves, but it looks more sort of like all or none or explosive events. And so we're interested in characterizing these patterns of spontaneous activity by looking a lot at experimental data recorded in vivo. And we want to understand what role they might play in the refinement or in the setting up of connectivity of circuits in development that ultimately gives rise to computations. And so I'm going to show you examples today of how we have addressed this. There was a keyword missing in my title today, but it was in the program across scales. And so something that we can do as computational neuroscientists is really pull together data sets and knowledge that different experimental labs have collected at the different scales and try to build kind of a unifying picture to understand how the spontaneous activity might be shaping and organizing connectivity in development. And so I want to first start with the lowest scale. I believe I'm the only speaker at this conference that will discuss synaptic inputs, I think. So let's look at spontaneous activity and the role that it might play in organizing synaptic inputs in development. So these were experiments that were done a little bit more than 10 years ago now for the first time in the Lohmann lab and also in 2 other labs in the world, and now other labs have replicated them, of course, that investigated the organization of synaptic inputs on individual dendritic branches. The synaptic inputs here are shown as these black dots. So this is a pyramidal neuron from the visual cortex. I believe it's layer 2-3 neuron. And so with these two photon calcium imaging experiments, the experimentalists literally imaged the activity of these puncta. These are these synaptic inputs in the presence of spontaneous activity in the period of the second postnatal week. I forgot to say, in rodents, eyes open around postnatal day 15. So for the first 2 weeks, these animals cannot see anything. And this is when this spontaneous activity is expressed. And so if these experiments were performed around postnatal day 8 to 10, so just at the onset of the second postnatal week, and what they quantified was the amount of coactivity or correlation, related to correlation, as a function of the distance of the synaptic pairs. And so what they found was obviously a very non-flat relationship, so this monotonically decreasing relationship that showed that pairs of synaptic inputs that are within 15 to 20 microns of each other have a very strong coactivation pattern, so they become coactive together, they show correlated activity. And this is not the case for synaptic inputs that are far away from each other. And they call this property synaptic clustering, as is illustrated here in my schematic here at the top. So what we see here is, these clusters that I've colored here by the 2 different colors. And what is interesting here, we call them functional, or they call them functional, because we still don't know what these synaptic inputs may code for in terms of visual features. So it's really they're just correlated together, and they express this spontaneous activity together. But manipulating spontaneous activity was shown to completely abolish these clusters, suggesting that indeed this activity plays a role in setting up the clusters. And so a few years ago, we started to work on this problem to try to understand mechanistically what are really these activity-dependent mechanisms that use these patterns of spontaneous activity that we saw are non-random, are manifested in different brain regions that could indeed organize these synaptic inputs. And so we constructed a model at this lowest scale, which is the dendritic branch. We put synaptic inputs using measurements that we found in the literature of the density. The inputs here are indicated again with the black dots. This is about 200 microns long. And then we sort of needed 2 important components to construct the model. So the first component was we needed activity to drive these synapses. And as I said to you multiple times, this is the spontaneous activity. So here we use the model of these retinal waves that we took from literature, because this allowed us to generate lots of data that we could use to basically train our networks or our synaptic networks to see the result, the effect on the synaptic organization. To convert these retinal waves into specific activation patterns on the synaptic inputs, we used the simple receptive field model. And luckily for us, around the time we started to work on this problem, Sonja Hofer's lab identified or measured exactly the receptive fields of single synaptic inputs, albeit in the adult mouse cortex, and found them to be these Gabor, to be organized as to look like these Gabor receptive fields. So there are 2 things that are important. They're localized in space. So the box here is degrees in visual angle. So you see that they're not spanning the entire box, but they're really localized, and they're oriented. So the blue and the red correspond to positive and negative regions, so that excite or inhibit the synapse, and you see that they're oriented, so you can draw a line between the red and the blue region, and you see that they indeed show some orientation. And so this is how we model them. So we basically took 2 Gaussians that were sort of elongated in space. If you look at them from the top, they are elongated, look like our receptive fields, and we characterize them by 2 parameters, namely the orientation as well as the center. And the center is important because it tells us where in visual space, so azimuth orthogonal, if you take degrees of visual angle, where in visual space each synaptic input actually looks in, like which visual part of the visual world it codes for. Okay, so then we needed the second component in the model, which is the activity-dependent synaptic plasticity rule that uses these patterns of activity, namely these retinal waves, to somehow organize or reorganize the synaptic inputs that were originally randomly distributed on this dendritic tree. I forgot to mention, you noticed that my synapses became colored all of a sudden from being black, and that's because when we build this model for receptive fields, we now indicate the orientation preference of each of these synapses by just a color code. And so this is the color wheel that tells us what is the preference of each orientation preference of each synapse. And so the plasticity rule that we used, in my lab, we work a lot with plasticity rules. We try to infer them from data. We try to study what types of activity patterns they may depend on, bursts or spikes or timing of spikes and so on. But here we really wanted to really look deep in the mechanisms that may underlie plasticity. And the same lab that identified the clustering, the Lohmann lab, identified also a molecular mechanism that could underlie a synaptic plasticity that uses these spontaneous activity patterns to drive the clustering. So the molecular mechanism they identified relies on this interaction of two neurotrophin molecules, BDNF, and then a developmentally prominent version, pro-BDNF, that are separately responsible for different aspects of synaptic organization or synaptic plasticity. So the BDNF binds to track B and promotes potentiation and stabilization of synapses, and pro-BDNF has the opposite effect to sort of depress and eliminate synapses. And what was really interesting is they found this activity-dependent molecule, MMP9, that's released in an activity-dependent fashion that can basically modulate how much BDNF and pro-BDNF you have, and therefore can regulate how much potentiation and depression you have in the system. So when we and so what we were able to do is actually to reduce this molecular model into a more phenomenological rule. So all of you here know about Hebbian plasticity and STDP, and so what we were able to do is to basically show that the synaptic plasticity here, the change in the weights, is indeed contains a Hebbian term and another term that we call heterosynaptic. So let me just explain each one of them. So the Hebbian term just tells us that if you have synapses that are nearby, that are basically co-active because just by chance they happen to have the same orientation, they get co-activated together, they activate the postsynaptic stretch of dendrite where they make inputs on, and because of the coincident pre- and postsynaptic activity, they increase the synaptic strength. So here we have the correlation-dependent cooperation or stabilization of synapses. Now, the second ingredient is the competition or the depression part. So this is where this pro-BDNF plays a role. And so here we were able to relate to basically extract the single constant parameter that depends on how much pro-BDNF you have in the system that allows synapses that are nearby, the ones that are co-active, but are actually tuned orthogonally relative to the active ones, to be depressed because they are not activated, but nonetheless they experience postsynaptic activity because of the spread of calcium postsynaptically on the dendrite. And this is a distance-dependent effect, so the farther away they are, the less they experience the influence of the calcium. And then we added a structural plasticity ingredient because, at least at the beginning, when there's a lot of pruning of the wrongly placed synapses, we got a lot of elimination of synapses. And so this just made sure that we don't have an empty dendrite without any synapses. And so whenever a synapse is pruned, a new one is born at a random location on the dendritic tree with a random visual preference and a random orientation. And so with these two components, so we have the spontaneous activity that drives this plasticity rule, we generated indeed clustering. So this was, we generated in these clusters. So we simulated the model for 14, 15 days of development, just like in the visual system. And we saw that from this random organization of synaptic inputs, we converged to a scenario where we have these correlated clusters. And so indeed, we can compute the same correlation as a function of distance, and we see this decaying relationship as in the data. Now, what the model allowed us to do was actually really quantify how much correlation do we need. And so we wanted to understand, are we just lucky? Is this really like a fine-tuned point that the retinal waves have this right amount of correlation? And so the model allowed us to really explore the range of correlations that is possible and to sort of make predictions for what type of activity patterns, these spontaneous activity patterns, need to be in order to generate the clusters. And indeed, if we just simulate the model with noise, we basically completely abolish the clustering relationship. So it is an activity-dependent phenomenon. But then what the model also allowed us to do is to try to link these effectively developmental effects, because we were really inspired by the developmental literature, to what people have found in terms of clustering in the adult animal, because people have looked at different species, for example, in the ferret, and have also found this type of clustering, but now not just in terms of just spontaneous activity, but they could actually correlate it to a specific visual feature. So the lab of David Fitzpatrick in Florida was able to find that there is indeed clustering according to orientation in the adult ferret visual cortex. And this very nicely matches to what our developmental model predicts, presumably arguing that maybe this is a developmental phenomenon that either remains stable or gets somehow maintained in adulthood. However, if you're paying close attention, you may notice that the developmental data that I used to motivate the model was about the mouse, and now I'm comparing this to adult results in the ferret. If one actually looks at the mouse visual cortex, it seems like one does not see this orientation clustering that our model predicts. And so, so it's there in the ferret, but not in the mouse. And so what is going on? So how can you have developmental functional clustering in the mouse, but not have this orientation clustering in the adult mouse dendrites? And so there's many differences, of course, between the mouse and the ferret. And one of the things that we thought could be important here is actually the relative sizes of the cortices, which then determine sort of the amplification factor. So how how densely a particular set of inputs looks at or codes for visual space. And so it seems like the sizes of the cells and the dendritic branches are similar across these two species, but area-wise or length-wise, it's a difference of about 5.5, factor of 5.5, but area-wise, the mouse cortex seems to be about 30 times smaller. And so what this means is that you have a much bigger amplification factor in the ferret than you have in the mouse. And when we take this model into account and just leave the similar activity patterns as well as the similar plasticity rule in our model, but just change effectively what we did was we changed the effective spread of the receptive field. So this is what we had for the ferret. We have receptive fields that are really clustered in space. This is visual space, degrees in visual space. And in the mouse, they were really much spread in space to account for these differences. What we found that indeed, in the ferret, we do predict the orientation clustering, but in the mouse, what we basically predict is something that we call overlap clustering. So what we see are these gabors. Maybe it's actually easier to look at the colored ones here. Here I use, for the red and the blue, I just use full and dashed lines, but it's the same thing. In the ferret, we see that they line up. So the nearby synapses have the same orientation, but in the mouse, you can have nearby synapses that have almost completely orthogonal orientation, but nonetheless, the positive regions of these Gabor receptive fields lie on top of each other. And so this is sufficient to generate high correlations. So these synapses are indeed coactive. They're nearby and they're coactive. Even though you don't have this orientation clustering. And so, we could play this game more further. We also looked at the sizes of cortices of other species, the rabbit, the cat, and the macaque, and we could sort of generate this space. We also allowed for slight change in receptive field size, which we found to be not so important, as you can see from this plot, but we could kind of predict in these other species whether we would have orientation or overlap clustering. And I think that around the time that our paper was in review, there was indeed a study in the macaque that also found the same orientation clustering that we see, that we would predict and that we see in the ferret. And so we then continued and looked at these effects, not just at the local level, so not just at the formation of these local clusters on these small dendritic branches, but we could also study what this means for the global organization of synapses on the entire dendritic tree of these neurons. But I'm going to skip that. If you want to know more about this, please ask me at the end. I'm going to change gears a little bit and change scales as well, but still stay in the hypothesis that these spontaneous activity patterns in development really drive organization and refinement of connectivity in a way that generates computations. I will get to computations soon, I promise. So now we amplify the scale a bit. So instead of looking at the organization of these synaptic inputs, we started to look at the circuit level. And for this, we analyzed some relatively old data by now, but these are data sets that are just becoming richer and richer every day as the experiments are ongoing, that looked just at the visual cortex, so 2-photon calcium imaging of population activity, so individual cells in the visual cortex, and that identified, just by looking at the visual cortex, 2 different types of events that actually seem to correspond to the retinal waves versus endogenous events generated just in the cortex. So they're both manifested in the cortex. One, we call them local events, although the original study, they were called L events or low synchronicity events, because they activate very few cells in the field of view. These are colored here in red. And if you look at the population trace, you see that for the entire population, they're characterized by these low amplitudes. And then the second class are these global, or also called H events or high synchronicity events, because they activate many cells in the field of view. And in the population trace, they're really manifested with these large, these are really these explosive events that activate almost the entire network. And what was interesting is they were able to, by doing simple perturbation experiments, for example, removing the eyes, they were able to find that the source of these 2 events that have these clear statistical differences or features in the cortex is different. So these local events are actually generated in the retina. So they're the retinal waves that get propagated to the cortex. And these global or high synchronicity events are intrinsic. So they're just generated in the cortex without the input from the retina. And so we now wanted to look at very simple feed-forward models that take into account these spontaneous activity patterns with these 2 different sources to investigate the refinement of what we call topographic connectivity. Again, this has been a problem that dates back to people many decades ago. But here we build a network with just these 2 network layers, so a periphery layer and a cortical layer. We generated or we activated the cells in both of the layers using these spontaneous activity patterns. And so we didn't, so I'll show you in a moment how we solved the problem, but in this first initial study, we did not generate the spontaneous events ourselves. We just looked at the data, characterized the distributions of the amplitudes, durations, and frequencies, and we sort of use that data to come up with ways to stimulate the neurons in our network. We were mainly interested in the refinement, the topographic refinement of connectivity, trying to preserve neighboring relations from the input to the target layer. And so here, based on what we derived already at the subcellular level, we use the Hebbian rule, which basically says, okay, if you have pre- and postsynaptic activity, the change in the weight is just proportional to the product of the 2 activities. But as you know, this is extremely unstable. So in the subcellular project, we resolved this by having this pro-BDNF term, but here we wanted to keep it more general. And so the problem is well known. If the presynaptic activity drives the postsynaptic activity, the connection is potentiated, which drives more postsynaptic activity, which potentiates the connection even more. And so you have this positive feedback problem that many people have realized and tried to solve, including ourselves. So there's many ways to solve this problem, to add various mechanisms or that all exist in biology. Biology has come up with multiple ways to stabilize Hebbian learning. But what I want to argue today is that this stability, this mechanism to induce stability in the presence of Hebbian learning seems to be inherent in the type of activity patterns that is seen in this developmental model. So these are actually these global or high synchronicity events that we think are the ones that can maintain stability. And so initially what we found is we had a model where we eliminated the global events and we just looked at what role these local events might play in refinement of connectivity. And what we found is that actually it was the model, so we looked at the change in the weight according to the Hebbian rule, so we modified it a little bit to add this depression term that we had from the subcellular model. So basically, as a function now of the presynaptic activity, you can either have depression or potentiation relative to whether this presynaptic activity is smaller or bigger than this depression threshold. But we found that to get any kind of stability in the system, you really have to fine-tune this threshold parameter. And moreover, not do you only have to fine-tune it to get the refinement to work, but you sort of constantly have to adjust it throughout development. So the model is very sensitive, and nobody wants sensitive models. But then we realized that actually, rather than doing this by hand, simply the presence of these global events can adjust this threshold and therefore modulate how much potentiation and depression you could have in the system. And indeed, this can be done in a very dynamic fashion. So actually what we predicted was that these H events, when they are generated, these global events, when they're generated in the cortex, they're generated in such a way that the amplitude is somehow senses. So there's some kind of sensor that keeps in track the activity in the network before the generation of these cortical H events, these global events, and then adjust the amplitude of the newly generated event in a way that is proportional to the ongoing activity sometime before. And so indeed, we analyzed the data and we found a signature for this correlation, suggesting that these global events can really endow this Hebbian learning in the network with stability by sort of dynamically regulating themselves to the ongoing activity in the network. And so when we now added this adaptive property, this stabilizing property that is there because of these global H events, we could indeed go from an early scenario where we had this all-to-all connectivity from the sensory to the target area with very weak and diffuse connections into a very well-refined scenario with these receptive fields that are topographically organized where the connections are strong. But not only this, what we could also predict was what would happen to the activity as a function of development, because usually we kind of think always of, okay, activity as is it being instantiated by synaptic plasticity affects circuit organization because it modifies the synaptic connections according to these learning rules. But then as the network is changing and is modifying the connections, obviously it will affect the activity that it generates, especially these global events that are intrinsic to the cortex. And indeed, we saw that as we had these global events that dynamically adapt to the ongoing activity in the network, we found that they can also account for the developmental sparsification of activity as a function of age. So, of course, in our case, it was as a function of simulation time in the network. So that in early developmental stages, we see these very sparse but prominent large events. And as a function of age, they become eliminated and get replaced by these small events. So this is around eye opening, which makes total sense because you don't want to have, at the time of eye opening, this extremely epileptic tissue that generates and activates the entire network, but indeed starts to look more like sensory-driven activity. And so we're now looking at ways to kind of make this even more interesting and more biologically relevant. So rather than putting in these activity patterns that we measure from the data, we now wanted to think about mechanisms by which these activity patterns could actually be generated by the networks themselves. So we are, of course, the networks are not feed-forward. So now we're considering neurons, excitatory and inhibitory, which we before we were a little bit ambiguous. We sort of only modeled excitatory neurons. Now we acknowledge that there's also inhibitory neurons and a lot of recurrent connectivity between the excitatory and the inhibitory populations. So we have a network as a model of the visual cortex, and we also have these spatiotemporally structured spontaneous events that come from the retina in the form of retinal waves, as well as a mechanism to generate these global events in the cortex. And so our current work indeed has succeeded at generating both the local and the global events. And so this is an example of a local event. So here the high red indicates a very high firing rate. So this is sort of like our visualization of what would be a calcium imaging experiment. And so what we think the important part in how these local and global events are driven or regulated is inhibition. So these local events are the ones that come from the retina through these retinal waves, and they simultaneously activate. So they go both to the excitatory and the inhibitory cells. And because they activate the inhibitory cells at the same time as the excitatory ones, they prevent the global spread of these events. And so you don't have these big explosive events. On the other hand, the global ones, we have some intrinsic mechanism where a fraction of these pyramidal cells is spontaneously active, and recurrent connections which amplify this activity. And therefore we have these large events. They are eventually killed by the recurrent inhibition, but this is delayed. So there is this window of opportunity when these global events can indeed be generated because there's a slight delay. So there's this disynaptic connection. First it has to get to inhibition before it comes back to excitation to prevent the events from spreading. And so now we're trying to combine these two models. So a model where we have the generation of activity and a model that we have the plasticity to kind of combine the whole picture together. And just a small side note, we're also interested in characterizing how this spontaneous activity may differ in different type of, in this case, in a disease model, because there's, of course, changes in just from wild type animals. And so here we're looking at the spontaneous activity patterns in a autism mouse model, Fragile X, which has a single gene mutation in the FMR1 gene. And so what we see here, just broadly, if we just characterize these local and global events, normally we have a predominance of the local relative to the global events, but in these knockout animals, this ratio seems to be equalized. So that you actually see quite a few of these global events that are really synchronizing over the entire population. And we're testing two alternative models where either the feed-forward or the recurrent inhibition is perturbed to identify which model could actually account for the changes in the relative ratio of these local and global events. Okay, so I'm going to stay at the network level, but now I'm going to move a little bit away from development, and I'm going to tell you about computations, because we know I think it's really fascinating to study development and to look at how all these things have to come together to self-organize the circuit, but ultimately these circuits have to do something. And so, and a lot of what the circuits do, I think, is really driven by not just the feed-forward inputs they receive. They don't just need, circuits don't just need to process and transfer information. They actually have to use it in some way to do the computations. And so this is just to tell you what we already realized with the model of spontaneous activity, that cortical circuits and many other brain circuits are recurrent. This is a CAT V1 showing anatomical connectivity, but this one of many experiments, this is from Massimo Scanziani's lab, that basically shows that it's not just that anatomical connections are recurrent in these circuits, but we also have a lot of the functional importance of these connections, of these recurrent connections, is also big. And so here is a normal experiment where they patch a cell in the cortex, and the cell received normal feed-forward and recurrent input. And so you can measure the total amount of excitation in the red curve here. And then with a simple optogenetic perturbation, one can eliminate all these intracortical inputs from the recurrent network. And one can see that what is remaining basically from the feed-forward stage, which is much less than the total amount of excitation, really arguing for the contribution of these recurrent effects. And so the computation we wanted to study, where we know specifically that these recurrent interactions play a role, is known as response normalization. So this was originally introduced and characterized by Laura Busse and Carandini and Harris at UCL, but now has been reproduced in many other labs. And so it's a very simple experiment where they look at the activity, in this case of the visual cortex, the population activity, when they show different oriented stimuli. So when they basically, first they do a baseline experiment to characterize the baseline activity, and then they show two gratings at different orientations, one at a time, and measure the population response. So here the grating is horizontal, here the grating is vertical, population shows a peak at the different angle. But the surprising thing was that when these two stimuli were combined in this flat grating stimulus, the total response was actually sublinear. So it was less than the sum of the two individual responses. Moreover, if one of these stimuli was shown at a lower contrast than the other, then the network response was a winner-take-all response, meaning that there was one, the more prominent stimulus had the big response, and the less prominent stimulus had basically a suppressed response. And so this is known as response normalization. And one prominent model in recent years that has been proposed and analyzed by Ken Miller at Columbia and people who have come from his lab has been the so-called stabilized superlinear network, or the SSN. I'm not going to go into details about this network. The key property is that the network is recurrently connected. So you can generate this response normalization because the network is strongly recurrently connected. So you need excitation and inhibition to really influence each other. And the second important component was that the neurons have nonlinear response functions as a property of, as a function of the input. So if you provide input current, then you see that the network shows a nonlinear response, which again, I think many people find and would agree with. And so, but in all of this analysis that people have done, they have always used sort of hand-tuned connections. So they've always said, okay, we assume, we know it has to be strong, but exactly how you tune the parameters has always been somehow arbitrarily decided. And so with one of my PhD students, Sam Ekman, we really wanted to kind of tackle this difficult problem. Can we achieve a scenario where we can self-organize the connections in this network that can capture this computation known as response normalization? And so we started with a completely plastic network, so excitatory inhibitory neurons, the triangles and the red circles. These light blue circles are basically the input units. So here we're not talking about spontaneous activity, but inputs in the form of these visual inputs, which are the gratings. And we basically assume that all the connections, recurrent as well as feed-forward from the periphery, are plastic, meaning that they can be changed according to the activation from the activity that is presented feed-forwardly with the plasticity rules. And for the plasticity rule, we took the classical Hebbian rule, and we know this is unstable, but the situation is even more complex when you add recurrent connections. And so again, as I said, biology has come up with multiple ways to stabilize Hebbian learning. So here what Sam proposed was a mechanism that probably all of you have used and are aware of. It's just normalization of the synaptic weights, or synaptic scaling. In other words, whereby if you have two synapses and one of them undergoes LTP, that synapse of course becomes potentiated, but this of course has the problem that the synapse can grow really big and the network can be unstable. And so simply by applying normalization, by scaling down in a multiplicative way to the two of the synapses, one can stabilize the system. So you still can preserve the fact that the stimulated synapse is stronger than the non-stimulated synapse, but you overall preserve the total amount of synaptic weights. And now the key insight that Sam had was to apply this both to the excitatory and the inhibitory cells. So basically the connections into an excitatory neuron get normalized separately from the connections that go into an inhibitory neuron. And he was motivated by a pretty landmark study from Bourne and Harris some years ago that basically looked at electron microscopy after the application of LTP for both excitatory and inhibitory cells. And I realized the inhibitory data is a little bit sparse, but nonetheless it's there. That indeed showed that the total synaptic area is preserved before and after. So it's the same before and after the induction of LTP. And so to study this in the fully plastic network, as we have here, we did this by basically analyzing different circuit motifs, and by focusing first on the feed-forward and then the recurrent connectivity before putting it all together. So let me take you through some intuition. I'm not going to show you the math, but so what we did here is assume, so this is one motif with two excitatory neurons in the cortex that receive feed-forward inputs that are seeing these oriented gratings. So here, this pyramidal neuron is plastic. So these dark connections, these feed-forward connections that it receives are also plastic. This is indicated here, synaptic weights. So you see, initially they start being completely flat. So this neuron at the moment is totally non-selective. It doesn't really respond more strongly to any one orientation than any other. But we have another excitatory neuron. This one has fixed connections, and it is orientation tuned. So it really likes to respond to stimuli that are oriented at 135 degrees. And here we were very interested in the role of this single recurrent connection between the excitatory cells. And so what we find is that these excitatory recurrent connections have what we call an attractive role. So we start with a random distribution of these synaptic inputs, of these synaptic weights, and we see that due to the presence of this recurrent connection, the tuning curve of the feed-forward weights of this plastic neuron becomes attracted to the other one. And then we did the same thing with an inhibitory neuron. Actually, we have more than the one that is shown here. So we have several. We distribute them uniformly through orientation space, but we begin again with a plastic excitatory neuron that is untuned, but it can tune its curve subject to plasticity. And if you had to guess, if the functional role of this connection from the excitatory cell is attractive, then the role from this inhibitory connection will of course be repulsive. And that's indeed what we see. The tuning curve feed-forwardly towards this excitatory neuron becomes repelled from the other ones. Then we make the circuit more complicated. Here we actually have multiple excitatory connections, and we have two tuned excitatory cells in a feed-forward manner. But because the recurrent connections are so powerful and strong, the effect that they attract each other is just so potent. So the recurrent connections actually dominate over the feed-forward ones, and the feed-forward tuning aligns so that they have the same orientation tuning. And similarly, when we have inhibitory ones, they actually end up repelling each other and completely decorrelate and cover the entire stimulus space. Okay, so then we can combine all of these effects, and we can develop a theory that allows us to study what happens in a fully plastic network. And so let me now analyze for you what are these excitatory feed-forward. So the feed-forward connections to the excitatory cells, they all begin flat. So these are the inputs, the weights from the inputs into the pyramidal neurons. And these are the feed-forward connections, so from the inputs to the inhibitory neurons. They all begin flat, so we have no tuning in the excitatory and the inhibitory cells. But we see that in this self-organizing mechanism, they all achieve a state where they completely distribute uniformly across stimulus space. This happens simultaneously to this recurrent plasticity, so the recurrent weights, which are the important ones, which are also plastic. So let's look at what happens to those. So here we can basically line up all the excitatory cells and all the inhibitory cells. And so this big matrix gives us the recurrent connections between individual excitatory cells on the main diagonal. This little matrix also on the main diagonal gives us the inhibitory to inhibitory connections, and then the off diagonal gives us the cross connections between the inhibitory and excitatory neurons. And what you can see here, so here the dark color indicates strong weight. You see that these matrices are block diagonal. So what that means is that neurons, we have ordered them here according to their feed-forward orientation preference, right? According to their tuning. So neurons that are similarly tuned, that will maybe have tuning curves that are next to each other, would also have a very strong recurrent connection. And this has now been found in many different sensory cortices by many labs. And this is something that comes out almost as a given from the self-organizing mechanisms in our model. And finally, so we are indeed able to train this network, as I promised, to compute to produce this response normalization. So if we now show two gratings at a time, one at a time, so just the orange, the cross 45 degrees or the minus 45 degrees, we see responses of the network shown here in orange and green. And when we show them together, we see this sublinear response that is smaller than the sum of the original responses. And similarly, when we reduce the contrast, so the amplitude of one of the stimuli of the gratings, we see this winner-take-all mechanism whereby one of the stimuli really dominates the response and the other one gets won out. Okay, so the other thing we can do is we can, of course, modulate the contrast of the second grating. And so what we see, just like in data, we actually see a divisive effect. So when you actually change the contrast of the second grating, we see that this curve actually shifts in a divisive way, which looks exactly like the data. And so, yeah, we can capture basically other effects from this model. And the important thing is that it comes about purely from this self-organizing manner rather than having to fix and tune the connections ourselves. Okay, so I'm going to end by showing you actually some new things that we're doing now. So the conference is not just about computation, but maybe cognition. It's a little bit difficult to study cognition in developing animals or behavior, but we're starting to think about this. And so first we started to think about how spontaneous activity could be modulated by other things that go on in the brain. And one prominent thing that goes on in the brain, of course, is neuromodulation. And in development, and in one of the most important neuromodulators is oxytocin. So this is the love hormone, or whatever you call it, that is released, that is important for building maternal and child bonds. And so basically these animals, just like any animal, when you separate them from the mother, they tend to be in distress, vocalize, and seek attention, and they want to be close to they want to attract the attention of the mom and be next to the mom. And so again, in collaboration with Christian Lohmann, who's now my long-term collaborator, they first did these experiments where they just did an imaging experiment where they dumped oxytocin while doing imaging. This is a wide field imaging. They also did 2-photon imaging of cortex that includes not just visual and somatosensory cortex, but also some higher order areas here that I'm not showing, like RL and LM and so on. And what they found, surprisingly, was that the application of oxytocin significantly reduced the activity primarily in V1. It had less of an effect in S1, but it primarily reduced the activity in V1. So one can quantify this. This is the baseline. This is activity in oxytocin. You do not see this in the somatosensory cortex. And so I can show you many, many plots where we looked at this and confirmed this. So it is the frequency of the events that reduces, and it is also the correlations between the individual neurons. So somehow the network just becomes less active and less synchronized, the V1 network, in the presence of oxytocin. They were able to identify that one reason that you have these different effects in the visual and the somatosensory cortex is that there's really a lot of oxytocin receptor on a specific class of inhibitory interneurons known as somatostatin interneurons. And therefore, it seems like oxytocin ends up exciting these somatostatin interneurons. Because it excites the somatostatin neurons, you have a lot of inhibition in the system, and therefore the activity decreases, both frequency and correlation. And we were able to build a model to capture this effect. But we didn't end here. So in the problem always of the experimentalists was that this oxytocin was released in an extremely non-endogenous way. You basically go with the pipette, you put the oxytocin, and you see this big effect. And so a joint postdoc of ours had to come up with this really cool paradigm to release oxytocin in a natural way. So the mouse is being imaged. There's also a motion sensor that gives us some behavioral data, let's say. There's a camera where we can see what the animal is actually doing. And there's a microphone that records these vocalizations that the animal makes, because obviously this is an experimental setup. The animal is separated from the mother, so it's obviously super stressed out and is vocalizing. And so she designed this setup where the animal can actually hopefully get an oxytocin endogenously released in its brain by puffing air through a cage where the mother is sitting. And of course, there's the control where the air is just puffed through an empty chamber. And so we have some very preliminary results. I will show you. It's really more analysis at this point than a model, but we're very interested in sort of linking the model that we did have with the role of the somatostatin interneurons. Okay, so here's the neural activity that we have. Here's the movement of the animal. And I'm just going to play for you. You've probably heard this, but maybe this is not so important. These are, so the animals make two types of vocalizations. These are ultrasonic. These are ultrasonic vocalizations. So we have all this data. One can look at the frequency spectrum and can identify these ultrasonic vocalizations, which are really released when the animal is stressed out. So when it really wants to call its mom. And these wriggling calls that are released, we can't hear these, that are released when the animal is hungry. And so we're starting to look at what happens. We have some evidence. I still have 5 minutes. Okay. So we still have, it started at 9:20. So, okay. So we actually, this is very preliminary, but we're starting to see that as a baseline effect, females tend to produce more USVs than males, but this maternal order does indeed reduce the emission of these USVs. And so we think the experiment was successful in the sense that we are seeing, in some sense, the effect that we want to see. So in all animals, males and females, the presence of maternal odor reduces the emission of these USVs, which are these vocalizations they make when they really want to be close to the mom. But this is really work in progress. I just wanted to show you that we have this multimodal data set. So we are trying to do more than just look at neural patterns of activity and connectivity, and trying to sort of include, to our best knowledge, the type of behavior that these animals are generating in development, which are these vocalizations. Okay, so I'm going to end with this. So what I've shown you are sort of 3 parts where we look at how spontaneous activity instructs organization, both at the subcellular and the cellular level, and can establish these functional networks that can generate certain computations. The most important slide is to really thank my group, who have been all really amazing and have been with me. A lot of students actually left in the last year since the move of the group. So this is a big retreat that we had last year where alumni and new students came. So it was really great. And funding sources. And thank you very much for your attention. And I'm happy to take questions.